Purported immunoreactive myocarditis in the presence of the spike protein. Illustrations of the human heart and spike glycoprotein. Myocarditis is the inflammation of the muscular walls of the heart. Inflammation results in the reduction of the heart's capacity to pump blood. Myocarditis signs and symptoms include arrhythmia, palpitations, chest pain or tightness, heart failure, shortness of breath, and leg swelling or fluid retention. Causes consist of bacterial, parasitical, and viral infections, autoimmune diseases such as lupus, sarcoidosis, or some that are organ-specific, such as irritable bowel syndrome, as well as drug-related agents such as cancer therapies like immune checkpoint inhibitors, clozapine, as well as COVID-19 and smallpox vaccines. Messenger RNA vaccine-related spike proteins are foundational to the immune response. The spike protein glycoprotein protrudes from the envelope of some viruses, facilitating virion entry into the host cell via receptor binding to the host surface receptor and subsequent fusion of viral and host cell membranes. SARS-CoV-2 circulating spike protein may be causative of disease attributable to COVID-19 vaccines. Spike protein may cause the breakdown and internalization of ACE2 receptors, possibly being disruptive of the renin-angiotensin system and increasing inflammation, thrombosis, and vasoconstriction. It may stimulate platelets, damaging to the endothelium, with subsequent arterial and venous thrombosis. Vaccine-related lipid nanoparticle absorption by immune cells and their reintroduction into the bloodstream, and an increase in exosomes bearing microRNAs and spike protein possibly result in the aforementioned inflammation. A purported causal link between COVID-19 messenger RNA vaccination and myocarditis, immune thrombocytopenia, and impaired adaptive immunity has been postulated. Repeated messenger RNA COVID-19 vaccines lead to abnormally high immunoglobulin G4 antibody levels, which can result in the inability to neutralize spike protein, which may circulate for a minimum of 28 days, causative of subsequent immune suppression and the development of autoimmune disease, such as the aforementioned myocarditis. There are cell lines experiencing messenger RNA uptake productive of spike protein that express same on the cell surface, causative of such autoimmune attack. The presence of spike protein and CD4 positive T-cell dominated inflammatory infiltration within cardiac tissue is indicative of vaccine-triggered autoimmunity. Inflammatory cells such as eosinophils, lymphocytes, macrophages, and neutrophils in the presence of cardiomyocyte necrosis in the myocardial interstitium and epicardium typical of myocarditis has been observed. The main cause of mortality may be a sudden arrhythmia such as ventricular fibrillation or tachycardia. The sudden mortality of young male athletes post-COVID-19 vaccination is postulated to be caused by a catecholamine surge during physical exertion and the early morning wake-up period when the surge of epinephrine and norepinephrine occurs. Myocarditis is also believed to be a consequence of multisystem inflammatory syndrome, possibly attributable to systemic distribution of the aforementioned messenger RNA containing liquid nanoparticles subsequent to vaccination and consequential systemic spike protein expression and circulation with resultant extensive inflammation. Durable messenger RNA gene-based instructions within cardiomyocytes, pericytes, and other cell lines result in spike protein, cell surface, and interstitial space expression that incite the aforementioned autoimmune reaction. Myocarditis may be self-limiting or causative of severe or life-threatening disease. This figure illustrates the immunological mechanisms underlying the pathogenesis of myocarditis. The knowledge we have for understanding the cellular and molecular pathogenesis of post-viral and autoimmune myocarditis is substantially based on animal models. From these models, it emerges that the progression from acute injury to chronic dilated cardiomyopathy can be restricted to a process that includes 3 phases. In the first stage, acute injury promotes cardiac injury sustained by exposure to intracellular antigens such as cardiac myosin. The activation of the innate immune system activates the subsequent acute inflammatory phase, which develops over the course of several weeks. In this phase, specific immunity, mediated by T lymphocytes and antibodies directed against pathogens, and similar endogenous cardiac epitopes, is triggered, causing a strong inflammatory response. Although most patients succeed with the available immune defense mechanisms in eliminating the pathogen, a downregulation of the immune reaction follows with the development of minor sequelae. However, in other patients, virus clearance does not occur, leading to persistent myocyte damage. Heart-specific inflammation persists and tends toward chronicity, which is furthered by the misrecognition of endogenous cardiac antigens as pathogenic entities. Image of patients with active myocarditis that is suggestive of a lymphocytic and histolytic infiltrate and higher T lymphocytes in heart tissue sections. Hematoxylin and eosin immunochemistry of heart tissue samples depicts characteristic lesions of acute myocarditis with widespread lymphocytic and histolytic infiltrate, red arrow, and associated myocyte damage, black arrow. Evidence of SARS-CoV-2 spike protein in cardiac tissue after COVID-19 vaccination, A through C. Representative immunohistochemical stainings of SARS-CoV-2 spike protein in eosin methylene blue from patients with diagnosis of inflammatory cardiomyopathy after receiving Comirnaty, panels A and B, or Vaxzevria, panel C. D. SARS-CoV-2 positive cardiac tissue served as positive control. Inflammatory cardiomyopathy in response to COVID-19 vaccination is dominated by CD4 positive T-cells, A through C. Representative immunohistochemical stainings of CD4 positive and CD8 positive T-cells in endomyocardial biopsies from patients diagnosed for inflammatory cardiomyopathy after receiving Comirnaty, panel A and B, or Vaxzevria, panel C vaccines, respectively. Further determinative and confirmatory investigation, as well as peer review is indicated concerning the causality of COVID-19 vaccinee sudden and unexpected mortality from suspected myocarditis. Please refer to the other bibliographic references listed below each image presented and the following references. New presentations coming soon. Thanks for watching. Thank you to current and new subscribers.